Everybody, we'll go ahead and get started. I'm going to go ahead and turn off the lights in the front of the room so you guys can see the screen a little better. We can, yeah. All right, so you guys, today we're going to talk about kind of an interesting class of drugs, namely drugs that actually target DNA. So DNA is the target. And then on Friday, we're going to talk about drugs that target RNA. So nucleic acids for the rest of the week. These are some pretty important drugs, and they're very specialized because they do target DNA. So I went back and looked at Dr. Ineskevich's lectures, and you've had some content on DNA replication, the cell cycle, and probably also a little bit of knowledge about DNA structure, nucleotides, and the like. And so I think you're all set up with the background information, but I am going to assume that you know something about DNA replication in order to understand this lecture. But I'll try to explain things the best I can nonetheless, and it's an interesting thing. So what kind of drugs are we talking about that might bind DNA? Before I even get into this, I'll say that for the most part, you're going to hear about drugs that are antiviral drugs because viruses have genomes and viruses replicate very rapidly, right? Many viruses actually have DNA as part of their genetic life cycle, especially retroviruses that have an RNA genome, but that genome has to get turned into DNA by the polymerase reverse transcriptase. And so these drugs that basically mess up DNA replication are very effective antivirals. In addition, there are a few DNA drugs that are actually antibiotics, and they're messing up the DNA replication of bacteria, in some cases fungi. And then lastly, we're going to talk about drugs that target cancer. Because as you know, cancer cells divide very rapidly, and cancer cells have a genome that must be intact for cancer cells to stay alive. And so there are a lot of approaches that target cancer by messing up the cancer DNA. And these tend to be a little bit toxic because we as humans also have DNA, right? But since cancer cells divide so rapidly, anything that inhibits DNA replication in cancer cells is going to kill the cancer with the side effect of being somewhat toxic to the human. But cancer is serious, and so most people can tolerate a period of toxicity during chemotherapy to address cancer. So that's kind of where we're headed. And if we think about these types of drugs that target DNA, they fall into two classes. The first example are drugs that actually covalently bind to DNA. So there are certain drugs that actually form chemical bonds with DNA, and that will really mess up DNA replication because you have altered, chemically insulted the DNA such that it cannot be replicated. And alkylating agents are kind of case study number one for that, and we'll go into that. And then there are other drugs that do not bind and form covalent bonds with DNA, and yet they still muck up DNA replication. And these are drugs that can intercalate between bases. So they invade base pairs and stack within base pairs and disrupt DNA replication in that way. And then there are some drugs that can actually bind to the grooves in DNA. So the bases, even in an intact double helix strand of DNA, are accessible through the major and minor grooves, and we'll show you some examples of that. I think the other thing I'll mention is that you're also going to see today, it's not shown on this slide, but you're going to see that analogs are very important here again. Nucleotide and nucleoside analogs. These are nucleosides and nucleotides, which have altered chemical structure, but they look like nucleotides, and therefore DNA polymerase, for instance, will recognize these and try to incorporate them into DNA during DNA synthesis. So we saw that analogs are very important as enzyme drugs. You're going to also see that analogs are very potent drugs to inhibit DNA replication. Okay, so first let's talk about these drugs that actually form covalent bonds with DNA. And so these are covalent ligand-target interactions. And these are weird drugs because most drugs don't covalently bond to their target, but in this case there are certain drugs that do. They cross-link with DNA basically. So a lot of cancer drugs are like this. They can form a covalent bond with DNA. We'll go through a couple of examples here. Nitrogen mustards. Melphalan is one example. And another prominent example are platinum complexes, and kind of a famous one here, cisplatin. So these are alkylating agents. That means they add a small organic addition to DNA. More specifically, they tend to target nitrogen 7 in guanine bases, and that happens to be exposed in the major groove of DNA. So the DNA doesn't have to unzip for these drugs to work. They can just come along and chemically modify DNA, adding these small alkyl groups. And once that happens, DNA polymerase can no longer replicate that DNA. So what are these drugs doing? We are intentionally damaging DNA such that it cannot be replicated properly, which is kind of a risky thing to do because it can also cause mutations. So one of the caveats of this is that drugs like cisplatin, nitrogen mustards, all these alkylating agents, they do indeed work very well on cancer because they mess up the cancer genome by mutagenizing it very severely. But they also have a tendency to mutagenize the normal genome in our cells. So some of these cancer-causing drugs are carcinogenic, strangely enough. So you can treat a cancer with these drugs, but there's risk of later cancer that's induced by the drug itself that might occur 20 or 30 years later. But you know, if you're going to pass away, succumb to the cancer in a year, if nothing is done, that's a trade-off that's worth doing. But do be aware that many of these drugs also just cause mutations in and of themselves because of their mode of action. So they tend to be quite toxic. They target cancer cells. And the reason they target cancer cells is that cancer cells divide probably 10 times faster than any other cell type in the body in somebody who has a tumor. So how does this work chemically? Actually, there are a lot of, these are the 4 bases in DNA: thymine, adenine, guanine, and cytosine. These are purines. These are pyrimidines. And you can see that the nitrogen atoms in all of these bases are sites at which alkylation can occur. So the nitrogens are especially involved. And if you look at a base pair of DNA, you will see that here's, for instance, an AT base pair. Here is a GC base pair. And here we're looking down the axis of the double helix, viewing a base pair kind of from above. And you can see that in the double helix, there is a major groove and a minor groove. But the point here is, and it's a little bit different for an AT base pair versus a GC base pair. But the point I want to make is that the major and minor grooves allow base pairs to be accessible to chemical agents that come in from outside of the DNA double helix. And you'll notice here that some of these nitrogen atoms that are subject to alkylation are exposed in the major and minor groove. And so that's how an alkylating agent gains access to the bases in DNA. So these alkylating agents actually covalently attach themselves. They form bonds with bases. And it's an irreversible chemical addition that's going to really mess up the base pairs, right? And once this occurs, it interferes with normal functions of replication. Most notably, enzymes that replicate DNA cannot handle these alkylated DNA strands. What does that mean? It means that they can't replicate the DNA. So if a virus, for instance, is trying to make a copy of its own DNA or make a cDNA copy of an RNA genome, which is what reverse transcriptase does, it cannot proceed because the chemical is all, the DNA is all modified through these alkyl groups. So another thing that kind of plays into this is the nature of the type of chemical modification that occurs. There are DNA repair mechanisms, which we have in all of our cells, and that is kind of cleaning up spontaneous rare mutations that are always occurring during DNA replication. But if you have an alkylated base, one of the things that can happen is that there are repair functions that will just simply delete the altered base, and then you have an abasic site. You have the sugar phosphate backbone intact, but no base. And if another base is not put in there through DNA repair, that is a base pair that's now missing a base. So you can't actually, there's no possible base pairing during DNA replication. And polymerases will stall there. It can also get incorrectly repaired to make mismatch mutations. And in some cases, you can even lead to strand breaks, and DNA polymerase cannot handle broken DNA. But as I mentioned, alkylation itself, by its very nature, can also cause mutations. So that's the sort of game we're playing with alkylating drugs. Here is an example of what alkylation looks like. Here's melphalan as an example, and you can see the drug is very simple, has a couple of chlorine atoms in it, and it can form an intermediate binding with DNA. But at the end, and you don't have to remember exactly how this works, but what you can see is that this drug actually gets cross-linked to DNA. So it puts a small alkyl group addition to DNA, and it's going to happen specifically on guanines in this case. And so you can see that there's a kind of an organic chemistry mechanism. It's a nucleophilic attack of an iminium ion, but it results in adding an alkyl group to nitrogen 7 of guanine. So let's go back. Here's guanine, and nitrogen 7 is right here, so it's going to stick something to that nitrogen. And that's no longer a guanine that can form proper base pairs, and DNA, any polymerase will get stuck and stalled on that, and you'll not be able to continue DNA synthesis. So there are some of these DNA modification mechanisms vary between different alkylating agents. So for instance, it's possible for cisplatin to actually form cross-links, and melphalan as well, but this can either be intrastrand or interstrand. And so let's look at the consequences of this. Here are the types of mutations that occur from alkylating agents. You can get monoalkylation events, and that's where you have a single base, oftentimes a guanine, that has an alkyl group stuck to it. We would say in chemical terms it is an adduct, okay? And here's a very severe mutation. This is cross-linked between both strands. So you know that DNA has to separate in order to be replicated. this strand cannot separate. This double helix cannot separate because of this cross-link between strands. And DNA polymerase doesn't know what to do with that. It just stalls and replication is stopped. So the viral genome, for instance, or whatever's being replicated here cannot be completed. Even intrastrand crosslinks are a problem. Here is an intrastrand crosslink between two adjacent bases. And again, polymerases cannot proceed through such severe mutations. So the net effect of this is that when DNA contains these alkyl adducts, an adduct is just a small molecule that has been physically, chemically attached, it leads to a failure of DNA synthesis, and that can do one of two things. A common thing is that you'll get cell cycle arrest. So the cell is trying to go through its cell cycle, and it arrests. And that can also even trigger apoptosis, and the cell will kill itself, right? So it's great to induce apoptosis in cancer cells because you're destroying the tumor. Let's see. You know a little bit about the cell cycle also, because you've already seen this in this course. Where does DNA synthesis happen in the cell cycle? What phase? S phase, right? So what kind of an arrest would you get with an alkylating agent? It would arrest in the S phase of cell cycle, right? That's when the DNA synthesis is happening, okay? So oftentimes, if you look at cells that are treated with alkylating agents, they arrest in S phase. Simply because that's where the DNA replication occurs. Okay, so let's now get into some of these non-covalent ligand-target interactions. Questions on that so far? So these are very strong drugs that just physically damage DNA by cross-linking to them. And they're very potent anti-cancer drugs, but they're quite toxic to the human also. What about non-covalent? So there are other drugs that target DNA that don't physically bond to DNA, like the alkylating agents. And these associate with DNA via non-covalent interactions. And these include van der Waals forces, hydrogen bonds, electrostatic interactions, even altering hydration, presence of water molecules at the DNA binding site, and that's kind of a feature of how they associate. So let's go through some of these. So here is an antibiotic, actually, distomycin, and it binds in the minor groove, and it forms hydrogen bonds and hydrophobic interactions. So here is what distomycin looks like. And here you can see a double helix, and this area right in here is the minor groove. And you can see that distomycin can just kind of sit down in this minor groove, and because of these non-covalent interactions with the DNA, it just kind of sticks to that minor groove. And it forms a fairly strong association because there are extensive non-covalent interactions between distomycin and the DNA along the length of the drug. It just kind of sticks on there. It's like Velcro, kind of, okay? And so you can see that, for instance, it has some interesting features. The terminal amidine is positively charged. What is the charge of the sugar phosphate backbone on DNA? It's negatively charged, right? Because all those phosphates have negative charges. So this probably binds by a charge-charge interaction, the positive in distomycin, the negative in the phosphate, and then the rest of it just kind of sticks down like Velcro and associates with the minor groove. It has a preference for binding to AT-rich regions. And for some reason, this end here has some distaste, I would say, in terms of affinity for GC regions. But anyway, if a polymerase is coming along and trying to replicate this DNA and it encounters this, it's going to be really hard for that DNA to becomes single-stranded because distomycin is kind of gluing the strands together through a whole series of non-covalent interactions. So it's kind of like gluing the strands together, but with this non-covalent stuff. So that's an example of a drug that binds in the grooves of DNA. Here is another example, doxorubicin. And this is an intercalating agent. And so there are a lot of intercalators. What is an intercalator? It is something that can insert itself between base pairs. So it kind of wedges in between two adjacent base pairs and inserts itself, and then it interacts. And so it turns out that this is a phenomenon called base stacking. So there are actually, van der Waals forces are very important in interactions between successive base pairs. But this thing actually can insert between base pairs and bind that way. And so here you can see a DNA double helix. Looks like we've got 1, 2, 3, 4 base pairs shown here. And doxorubicin has inserted in between the first and second base pairs here. And since the, van der Waals forces are kind of weak, but you have so much of that van der Waal type interaction going on that this kind of, again, glues the DNA together, and it's hard for polymerase to proceed through that. Okay. By the way, ethidium bromide that many of you might use in the lab to stain DNA, that's another intercalating agent, and it's also mutagenic, and that's why you have to wear gloves and don't dump it down the drain, etc. Okay. Then there's this example, dynamycin A, which is another antibiotic. It's kind of an interesting thing. It's kind of a combination. Part of it can actually intercalate, and it also has some features that allows it to bind in the groove of DNA. So it's a little bit of a hybrid between the two types that I've already mentioned. This is an interesting one because it can associate with DNA through non-covalent interactions, but once it's activated by light or thiols or NADPH, and the last two can be present in cells, not so much light, it can actually cleave strands. So this is kind of an unusual drug because it binds by intercalation and minor groove association, and then it can be chemically activated and it will form a covalent bond with DNA that results in cleavage of the strands. Most of our cells are not exposed to light, but in the laboratory, you can use this to mutagenize anything that's growing in a dish because you can't expose it to light. But even inside of a cell in the middle of your body, which is quite dark, you can run into NADPH, for instance, and it can induce this cleavage event. Okay. So now we're going to spend most of the remainder of the time today talking about nucleotide analog drugs. These are quite important drugs, and they are really kind of famous for inhibiting viral replication, and they work quite well, actually. So these are actually synthetic nucleotides. So of course, nucleotides are the building blocks of DNA. ATP, GTP, CTP, TTP. So they're triphosphates. And an important thing to know is that a sugar and a base without any phosphates is a nucleoside with an S. A sugar and a base with 1, 2, or 3 phosphates, these are nucleotides with a T, and you'd have nucleotides monophosphates, diphosphates, and triphosphates. So only the triphosphate forms of nucleotides can be incorporated into DNA because that triphosphate has to be cleaved to incorporate into DNA. And the terminal 2 phosphates, the pyrophosphate, actually leaves, the diphosphate leaves. And so the first phosphate actually is incorporated into the sugar phosphate backbone of DNA. So this is chemically how DNA is synthesized. Now, it's important to note that all of our cells contain the ability to convert an unphosphorylated nucleoside into nucleotide triphosphates. And the way the cell does this is it adds the first phosphate, then the second phosphate, then the third phosphate. So nucleosides can be turned into nucleotides by progressive phosphorylation, yielding triple phosphorylation, which is an active nucleotide. So DNA polymerase, for instance, has to have nucleotide triphosphates in order to add bases. And so this is interesting because a lot of these nucleoside analogs are unphosphorylated as a drug. They have altered structure, but many of them are unphosphorylated. And what happens is they enter the cell through a nucleoside transporter, but then this nucleoside analog first becomes monophosphorylated, then diphosphorylated, then triphosphorylated by a series of kinases. So remember, kinases are any enzyme that adds phosphates, okay? So this nucleoside analog gets one phosphate via 5' nucleotidase, or I'm sorry, a nucleoside kinase, and then a second phosphate, and finally a third phosphate. And there are actually phosphatases and kinases that work. But anyway, the point is a nucleoside analog is actually a prodrug. It's a drug that has to be chemically changed in the cell in order to become an active drug. And in this case, the change that needs to occur is phosphorylation. And so all of this has to occur for bioactivation. So it's a prodrug that has to be bioactivated, and that occurs readily in cells. So people have made all sorts of modified nucleotides and nucleosides that serve as analogs, and basically human beings have played around with the structures of nucleotides and nucleosides quite extensively in search of drugs that inhibit DNA polymerase, basically. And so here is kind of an example of all the different things that have been done. Kind of a cartoon diagram of a nucleotide. We have the base, we have the sugar, we have phosphates. And think about, structure, function type stuff. Some of these drugs have azide groups, halogenated, things conjugated to the nitrogens in the base. Same goes for the sugar, halogenation, methyl groups. Even messing around with the ring structure of the sugar. And in addition, there are some drugs that have altered phosphates on them. So all three components of a nucleotide have been mucked around with chemically in the search of analogs that might inhibit DNA replication. So let's bring this into a little bit of biology, how this works, and we'll talk about antivirals. So some of the most problematic viruses are actually retroviruses, most notably HIV. And here is kind of the life cycle of HIV. It is a virus that is encapsulated, and it's bound in membrane. It has proteins in the surface of the membrane, and then inside the virus, you'll have the genome in the form of an RNA genome. When this associates with the cell via a protein called CD4, the envelopes fuse, the viral RNA is released into the cell, and then interestingly enough, the virus itself contains a molecule of reverse transcriptase. And this is an enzyme that we don't have in our own cells, because in human cells, you never turn RNA into DNA, but the virus wants to do this. So reverse transcriptase binds to the viral RNA genome, and it creates a copy of the viral genome using deoxynucleotides. And so it turns the RNA genome into, or the RNA genome into a DNA copy. And then that can be involved in replicating the virus. You transcribe it and make the RNA genome again. Or the DNA version of the HIV genome can get into the nucleus and actually inserts itself into the nuclear DNA as a provirus. So that's how it works. So there are nucleoside analogs that target reverse transcriptase, the ability of the virus to turn its RNA genome into a DNA copy. And that's how reverse transcriptase nucleoside analogs work. Yeah, and hepatitis B is another example of a virus rather like this. So these drugs are going to target reverse transcriptase kind of indirectly. What they're really doing is targeting the ability of reverse transcriptase to make DNA. So here is one example. There are many. And this is a nucleoside because it has no phosphates. So this is thymidine. And here is a drug. And what you'll notice is that, let's see, that's 1', 2'. At the 3' carbon in the sugar, it's modified. It turns out that hydroxyl and thymidine is very important because that's at the 3' position. You remember DNA replication happens 5' to 3'. This 3' carbon, this hydroxyl group ordinarily gets hooked up to a phosphate group in the nucleic acid backbone. In this case, it's blocked. So one of the other things that happens is this is a prodrug. So at this position here, the 5' position, because of bioactivation, this can get turned into a triphosphate. So it can get added to a growing strand. But once that's added to a growing strand, there's no 3' hydroxyl anymore, and you cannot extend that strand. Okay? So it incorporates and prevents elongation. And that's that's kind of a famous way to make an analog, is to replace that 3' hydroxyl with something else that cannot be polymerized. So it can get incorporated, but once it's incorporated, DNA replication is stopped right there. And it turns out that reverse transcriptase is happy to incorporate this nucleotide triphosphate analog once it's bioactivated, but once it incorporates it, it cannot extend further, and you halt reverse transcription, for instance, of the HIV RNA genome into DNA. Okay, so pretty interesting thing. Incidentally, reverse transcriptase is a very sloppy polymerase. It doesn't have much fidelity to it. Its only interest is polymerizing very quickly. And so this is a good feature because these drugs are less likely to do anything with endogenous polymerases because reverse transcriptase is so sloppy, it'll basically tolerate a whole bunch of mutations. That's why HIV changes genetically so rapidly, and you get variants. And so this kind of makes use of that feature of reverse transcriptase. Here's another nucleoside analog, idafovir, and it's another reverse transcriptase inhibitor. And here you can see, here's normal adenosine monophosphate in this case. But you can see that this one is weird. What's been changed? Well, the whole sugar has been changed, right? It's been opened up, and there's nothing resembling a ribose or a deoxyribose. This is actually a ribonucleotide because it has a hydroxyl group here at the 2' position. And you can see that the whole sugar ring has been simply replaced. But it still can become triphosphorylated by bioactivation. And this can get added, but then replication is over because there's not even a ribose there, okay? So it also has a modification that you can see, this phosphonate, in addition to being a chain terminating thing, also kind of increases its stability chemically. And so here's how it works. First, some bioactivation. It becomes triphosphorylated, and this is the version that will be accepted by a polymerase, and it can get added, but then there's no sugar, so you can't add the next base and the next base. It's just a chain terminating nucleoside analog. Make sense? So most of these nucleoside analogs work by preventing elongation during nucleic acid replication. Here's one that works on COVID-19 virus, remdesivir. Once again, it's a prodrug, and it's an analog of adenosine monophosphate. And so what you can see is that it looks like adenosine up here, except there's something weird going on there, isn't there? But at the phosphate, it has all these funky additions. But what happens is, when you put this into a cell, all this business gets removed, and then it can be triphosphorylated. And so it can get incorporated into nucleic acids, into the replicating viral genome. But then, because of some of the modifications here, it cannot be extended to make more nucleic acid. So it is a prodrug, and actually some of this business down here improves the stability of the drug when it's when it's introduced into the human, but it can be bioactivated. So just another nucleotide analog. So how do these work? They can also work for cancer. So cancer cells, and we're going to get into cancer stuff now, and perhaps some other types of drugs as well. But what I want to mention is that cancer cells do divide very rapidly. That means that during S phase, they're making a lot of DNA, and they divide much more quickly than normal cells. So anytime you block DNA synthesis in a cancer cell, you're going to arrest that cell in S phase. So it makes sense. If you block DNA synthesis, where do you arrest in the cell cycle? During S phase of the cell cycle. And there are all sorts of things that can cause DNA damage. We talked about alkylating agents already. UV light can do this. You can get thymine dimers. Ionizing radiation can do this, where you just damage DNA to the extent that it can't be replicated. And that's, radiation therapy for cancer. What you're doing is really damaging DNA. Chemical exposures, like alkylating agents, for instance. Anything that causes problems with S phase. So, normal cells, when they are exposed to some of these agents, they can kind of mess up DNA to some extent. But normal cells usually have pretty good repair pathways. So even if a point mutation is made in a normal cell, there are repair functions in the cell that can usually repair that. Cancer cells, because they divide so rapidly, a lot of cancer cells have just kind of disabled their repair mechanisms, okay? And so they're much more susceptible to modifications of the DNA. They don't know what to do with it. DNA repair takes time, right? And so cancer cells have eliminated a lot of their repair functions, DNA repair functions, because if they had them intact, they wouldn't be able to divide as quickly, okay? So although these anti-cancer agents do indeed damage normal cells have more capacity to handle that damage than cancer cells because of this reason. Just a little bit of review of the cell cycle. You already saw this with Dr. A, but here we have mitosis, and as soon as the cells have divided, you've made two daughter cells from a parental cell during mitosis. The first phase they're in is G1. They have a 2N complement of DNA. We say 2N because there are two chromosomes. Autosomes come in pairs, one from your father, one from your mother. Then DNA synthesis happens, and then you end up in G2 phase of the cell cycle. And there the DNA content is 4N, 4 copies of each chromosome. And then you go through mitosis and you reduce 4 back to 2, and you're back to 2N. So it's 2N through here. S phase is something between 2N and 4N. And then G2, you're in 4N, and then mitosis happens. So anyway, the DNA synthesis is happening during S phase. So these drugs typically cause S phase arrest. And then the cell can do one of two things. It either doesn't divide anymore, it just sits there, or this triggers apoptosis and the cell kills itself. And that's, by the way, a function mediated by p53, a lot of these famous cell cycle proteins. So let's talk about some anti-cancer agents. Fluorouracil. So this is uracil, a pyrimidine. It gets present in RNA because RNA has uracil instead of thymine. And here's fluorouracil, where we've replaced this oxygen with the fluorine. So it's halogenated, okay? And it's a pyrimidine analog. And in this case, it is just the base only. But the body can take bases and sugars and phosphates and put them all together. So it's possible for this to become a fluorouracil, for instance, nucleotide eventually. So fluorouracil has kind of a complex mechanism, and there's a lot going on with this. So here's fluorouracil. It gets into cells, and then there's all of this bioactivation stuff going on. It can become fluorine deoxy-UMP, and that actually inhibits another enzyme, thymidylate synthase. And so that's one of the enzymes that's involved in making thymidine, which is basically hooking up the uracil into a ribonucleotide. But even though it's a ribonucleotide, it can actually, this one has some altered ability to actually incorporate it into DNA as well. It can form into another modified nucleotide that causes RNA damage. So this thing can cause DNA damage and RNA damage. I'm not going to hold you responsible in an exam to know all this complexity, but what you're really doing with this drug is making a bunch of altered nucleotides that once again are going to work as nucleotide analogs. They're going to be able to get incorporated into, it gets incorporated into altered nucleotides that just mess up, in this case, both transcription and DNA replication. And once it's in there, because of the fluorine atom, it can actually cause a bunch of mutations. So you kill cancer with this big, huge mutational load that you induce in cancer cells. Here's another nucleoside analog, cytarabine, which looks like cytidine. And you can see, once again, that it's got a couple of different changes, very subtle in this case. This is a trans arrangement of the hydroxyl groups instead of cis. But that is enough to really alter DNA replication, and it damages DNA. So, you really have to have a normal nucleoside triphosphate, a nucleotide, in order to replicate DNA. And any subtle changes to the sugar or the base can really mess it up. But again, this is another example of a prodrug because it has to become triphosphorylated, and that's the 5' carbon right there. So this hydroxyl group is involved in, this is where the triphosphate gets attached, basically. So it has to be activated before it acts, once again. Gemcitabine, another example. What's the change here? 2 fluorine atoms have been conjugated to the 2' carbon. So it's halogenated. Once again, this has to be made into a triphosphate before it can be incorporated into DNA. But once it's in DNA, it can actually damage the DNA. And so let's look at what this drug does. How do you actually measure cell cycle? Well, there's a lab procedure or clinical machine called a flow cytometry. And what it does is you can get a whole population of cells and put them in a tube and pass them through a laser, one cell at a time. And if you have a stain in that cell that recognizes DNA, it measures how bright it is. So this machine can detect the difference between a G2 cell and a G1 cell. Because a G2 cell has a 4N complement of DNA, a G1 cell has a 2N complement of DNA. And you just count up how much fluorescence there is in a whole bunch of cells, and you can see how many are in G2 versus G1. And so here are normal cells, a whole bunch of cells in G1, and a number of cells in G2. And anything that's in between G1 and G2 is in S phase. And so you can see kind of in a population of cells, how many are in G1, how many are in S, how many are in G2. Well, here are the cells now treated with this drug, this nucleotide analog that causes S phase arrest, and all the cells come to pile up in S phase, basically, right here. There's still quite a few in G1, but there's a bunch in S phase, and G2 has been basically eliminated. So this is how you use a lab method, namely flow cytometry, which can measure the number of cells in G1, G2, and S. And you can see that the whole profile of the cell population changes, and they're stuck in S phase because they, and you can see that the consequence of that is they can't proceed to G2. And this is a typical pattern you see with nucleotide analogs. Okay, so what are some of the principles that we've talked about today? DNA is really a good drug target. And the reason mostly is that DNA replication is really important for cells, namely cancer cells. And it's also very important in viral replication. And so especially retroviruses that turn RNA into DNA. So DNA is a great drug target. You've seen that you can have some drugs that work in a covalent way by modifying the DNA, but there are other drugs that are non-covalent that can intercalate or bind to the major and minor grooves. And then notably, I guess it's not on this slide, but I think you should really understand the concept of nucleoside analogs. Remember, they have to be bioactivated, typically by phosphorylation. And basically, they can be accepted by a polymerase and put into a growing strand of DNA, but once it's incorporated into the growing strand of DNA, it's game over, and you can't make any more DNA downstream from that. And that's how they work. And so, minor chemical modifications to anything in the nucleotide structure can have these dramatic effects and make analogs. And I think you should also be aware that this is a bit of a dangerous game because, especially with the drugs that can chemically modify DNA, there's always a chance of inducing mutations in your own genome. So that's why these drugs get used for cancer, severe cancers, and not more minor things. Yeah. Any questions? I hope that all came through pretty clearly. They're really cool approaches, and they are based on, I think humans probably understand DNA replication almost better than any other kind of, biochemistry out there. And so our knowledge of how DNA replicates has yielded a lot of great drugs because of this. Okay, so we'll talk about RNA, siRNA, stuff like that on Friday. Thanks, and enjoy the rest of the day. Thank you.